Please join me in welcoming our next presenter, Dr. Vince Calhoun.
What is this here? Is that the easy button? Yeah, fire alarm? Okay, all right. Well, it's a pleasure to be here. What I'd like to do during my time is talk about data-driven fusion of multimodal data. So I'll basically start by going through a little bit of an introduction, kind of how I think about multimodal data and combining it, and just briefly touch on some of the many different methods that we and others have worked on. And then I'll cover some newer applications focused on one particular approach that we've found to be quite useful, and then talk about some more advanced models towards the end and end with some closing thoughts. So, you know, I think we're all kind of aware that, is there a way to get this moved? I can move that over there. There, does that work? A little better. I should see the title. Okay. So we know there's lots of different kinds of data, function, structure, connectivity, electrophysiologic data, genomic data. All of it is woefully inadequate to tell us what we really want to know, but what we are, so, you know, what we should be doing is trying to leverage the information across these different data sets and that is complementary. I always like to show this figure. This is my son. He's now a junior in high school, and he's doing data fusion when he was little. You know, what I hope we are doing is a little bit more sophisticated than what he's doing, and basically trying to leverage complementarity in the common example you can think of as EEG has high temporal resolution, millisecond accuracy. fMRI gives you localized hemodynamic information, and so bringing those two together makes a lot of sense. I think of data, combining data on sort of a spectrum here, where you start sort of on the left side, we have, you know, just doing two analyses and visually looking at them side by side, kind of fusing it in your head. Data integration would be something like PET-CT fusion, where you sort of co-register and put them on top of one another, but the data sets haven't really informed one another. Asymmetric fusion would be something like EEG-informed fMRI or vice versa, where one modality has benefited from the other, but not the other way around. And symmetric fusion, which is the highest information content, is basically decomposing them jointly and trying to leverage the information in both. And, you know, why do we want to do this? You know, maybe it's obvious, but I always like to remind us that, you know, so there are dimensions you can't see if you don't combine the data. So here we have just a simple example: brain function, brain structure. There are actually two groups in here, and you can't see a group difference, at least in the distribution, in either modality. But if we go through this direction here, you can see, you know, see the group difference. So just sort of a simple motivating example. Again, it's this joint information that's kind of invisible if we don't look at the modalities together. And I also like to look at the data in a multivariate way, and I think that's important. So we have a cloud of points here, and we can sort of pairwise link them together, of course. But if we don't use a multivariate approach, we might miss the fact that there are actually patterns that are linked within the data sets that then also are linked between the data sets in this case. So again, just a little bit of a motivation for us. And then lastly, thinking about it in terms of a carrot and a stick. So the carrot is, okay, if I collect EEG and fMRI, okay, it takes some extra work, but look I'll get these beautiful results, and I'll see, you know, networks, bilateral temporal lobe here, also in the EEG data. They'll match, I'll get great results, get more papers, et cetera. That's not always going to be true, but, you know, it's a very nice result. And then the stick is, well, you know, here we've got a result for two different stimuli for an oddball task, novels and targets. And so this is the fMRI result, and this is an MEG result in the same subjects for the same task. If you're an fMRI person and an MEG person and you write separate papers, they are not going to agree, right? You're going to get very different results. Which one's true? Okay, the MEG person would say MEG, the fMRI person would say fMRI, but I would say it's probably somewhere in between the two, because they're actually looking at complementary information. This is just a figure from a recent review we did looking at In this case, we were just looking at regression-based machine learning studies, so predicting continuous values from data. One of the things that we noticed was that, for all of those studies we could find that did multimodal, you see fairly consistent shift to the right in terms of prediction accuracy for multimodal data. So there's actually a value in multimodal data here. Another interesting thing, which is sort of a tangent, is actually we found that the patient values were actually more accurate than the healthy values. Here we're predicting things like symptoms, for example. Here we're predicting things like cognitive score and things you can assess in healthy individuals, suggesting those probably vary more subtly, than do the other values. It was actually a little surprising to me to see that, but it's quite significant. And so there is value in multimodal data. How do we start to combine it, right? It's all high dimensional. It's all very hard to do. One way that we like to approach it is through generating features from the data. And so what is a feature? Sort of a lower dimensional map of something of interest, right? So it could be an activation amplitude or a gray matter segmentation image or regions of interest, connectivity matrix, et cetera. Advantages are it summarizes the data, it's less computationally complex, takes advantage of all of the work that we've all done over many years. And so one of the things that we do a lot of is ICA decompositions, and I'll sort of take you through why we do that on a lot of different types of data, and sort of what has convinced us that it's useful. Here we're looking at sort of two different analyses. One was an fMRI analysis using a group ICA approach. The other one is ICA of, it was a task data set, and so we did general linear model analysis and got activation maps, and then did an ICA on those where we have subjects by voxels, right? And you get out networks from that analysis as well, right? So the intrinsic connectivity is influencing, or you could say biasing, those contrast maps or those task maps. This is work from a paper from Steve Smith where they're actually looking at just meta-level results. So coordinates from papers that were sort of smooth and then combined in the same way, and you ran an ICA on that. So that's even further distilled from the data. But you also get, you can also find these networks, from that kind of a decomposition as well. And what I'll be arguing is that these kinds of features are useful, and also when we do a lot of our data fusion approaches, we're actually leveraging this sort of inter-subject covariation, this information there. If you look at gray matter maps, brain structure, those also give you networks that look very much like resting networks, right? We could argue whether that's a bias in the T1 due to function, or whether it's actually a gray matter covariation that's meaningful and interesting and sort of underlies the functional information. If you look at sort of links between structure and function, you see strong decreases in age in sort of higher cortical areas like frontal lobe, and also you think temporal lobe regions. Whereas sensory regions, unimodal regions like visual cortex don't show such a drop-off. They stay similarly matched between function and structure throughout the lifespan. And this is a matrix showing the relationship between structural networks in terms of their covariation with one another. And you'll notice that this matrix is not noisy, flat looking. It's got some modularity to it. It's got some structure to it, which we've looked at in a number of different ways, and there actually is a lot of interesting information in there. We know that we can do this with fMRI, and we can also do decompositions of MEG data, where we're looking at sort of coherent networks in source space, reconstructed space. And then two more slides looking at diffusion information. So here we're looking at fractional anisotropy, and if we look at covariation in those maps, again, the full maps, you get out components that basically map different portions of tracks, right? And they map pretty well into some of the atlases, white matter atlases that you can pull from various places. You can go, again, a step back to capture some of the directional information. So if you compute, it's essentially a group ICA of diffusion data. So you have a voxel, all voxel to all voxel matrix per subject, and then you're basically decomposing those in a group ICA. And it basically gives you components that map very nicely sort of along portions of tracks. This is just a subset of the results looking at corpus callosum. And you have information in gray matter regions as well that can be leveraged. Okay, and I'm not going to be digging too deep into all of the different information that we can get out from that data, but we're using that kind of information in the subsequent slides that I'll be sharing. So in terms of methods, I'm not going to cover a lot of this, but here I'm just showing you there's a lot of matrix decomposition approaches that have been used. PCA, here's a subset of them, partially squares, canonical correlation analysis, joint independent component analysis, et cetera. I'll talk a little bit about this one just to introduce us to the concept, and then I'll talk a little bit more about this particular model. And so joint ICA is pretty straightforward. It's basically taking two different features. So maybe you have gray matter maps and you have functional maps, right? And you're basically identifying a joint maximally independent component that co-varies similarly across subjects, okay? And so the nice thing about this is that the features don't have to overlap. So you can have white matter regions and gray matter regions that co-vary that will appear in that component, or one modality might have some covariation and the other might not. You can, it captures all those different cases. Two examples of that, again, from some early results that really convinced me why we should be doing more of this, is here looking at fusion of two different fMRI tasks. We have a Sternberg working memory task and an auditory oddball task. If I'll just point you over here to the left, what I'm showing you here are joint histograms derived from the joint ICA output, okay? So it's basically auditory oddball versus Sternberg, right? And these are individual subjects, right? Top section here is patients, the bottom, patients with schizophrenia, bottom are healthy controls. And visually, you can see it's a pretty striking difference there in that the controls are sort of much more flat. There's less correlation between the two task sets of regions than in the patients where there's a significant correlation between them. So what is this telling us? It's telling us that the controls are sort of activating their brain to these tasks in a much more unique way. It's less correlated between the tasks. It's more specialized. And that's, I think that's pretty interesting to know. And it's something that, again, is completely invisible if you don't use two data sets and bring them together in this kind of a context. Another one that I like to show is where we have fMRI and EEG data that were collected during a task, but in separate sessions. So they weren't concurrent, so we didn't have to struggle with all of that, that setup time, or we just did it separately, so maybe it took twice as long, but nonetheless. And then we have two features. We have an fMRI task activation map and an event-related potential from the EEG data. And we're basically doing joint IC on those, and so we get an fMRI part of a joint component, and then the ERP portion, which is this blue curve here. And here's another example where we get another ERP and another fMRI map. And once you get that space decomposed, we have these joint components, and we have several of them. Then it gets interesting because then we can start to leverage the time resolution in EEG and sort of propagate that to the fMRI data. So for any given point in time here, we can take the weights corresponding to all of these different sources at that time point and reconstruct an fMRI map that corresponds to that, the combination of those weights. And it makes a lot of sense when we do it and play it as a movie. We get initial sort of auditory sensory, then we get motor planning, then we get primary motor, then we get this sort of large response around the P300 curve. And we've seen this in multiple different data sets. And it, again, it's kind of surprising, but you can actually leverage the high temporal information in EEG by virtue of sort of propagating through that covariation among subjects. And you can do the same thing in reverse, where you can pick a region in fMRI and reconstruct an ERP corresponding to that particular point in the brain. Just one slide also to say that with all these methods, you know, it's useful to kind of figure out if they're going to work, and also to be able to, you know, we need more advanced simulation frameworks to look at joint information, right? We can sort of make up things, but it's nice to have something that's sort of grounded in the statistical formulation so that we can relate them back to a ground truth. And so you can do that, you know, this is just one way that we've been working on it, where we can just simulate distributions, generate sources, and then estimate compared to that ground truth, and everything gets tied back to a statistical model for the two. And the one I'll talk more about and give some examples of is, and we're not just trying to add letters to acronyms and make things longer and longer and longer. We probably need to compress our acronyms, I guess. But multi-set canonical correlation analysis plus joint ICA, okay? We're going to add an R in there too for reference in a minute. But essentially, we're combining those two approaches together because they have complementary strengths and complementary limitations. So CCA is good at finding paired matches or multi-set matches between matrices, but it doesn't do well if it's not a unique match. And if it doesn't drop off quickly, then it's not going to find a unique separation. Joint ICA is leveraging higher order information, not just second order information. But when you do joint ICA beyond two modalities, it starts to get a little bit noisy because it can't find the links as well, as accurately. So we're combining these two basically as a first MCCA step, and then we're using higher order information with joint ICA to do the final decomposition. And we can add an additional constraint, which is quite useful at the front, which basically allows us to use something like behavior or symptoms or other variables of interest to say, well, find me a decomposed component from this data set, from this multimodal data set that sort of correlates with that, you know, with that result, with that behavior. And I'll show examples of that, so it hopefully will make more sense when we look at it. And I'll start with an ECT study. So this is some work by Jing Sui, who and Chris Abbott. So we basically have about 120 individuals who've had MRI scans pre and post ECT. And so these folks have major depression. And we have an fMRI data set and a structural MRI data set. And what we're doing is essentially MCCA plus joint ICA, but adding a symptom constraint, basically the depression scale that was used. So we're finding, we're only looking at this one component that comes out that is correlated with this depression scale. So the decomposition was influenced by that depression scale. Then we get the maps and we reconstruct the same information in healthy controls and compute a difference map. So that it's relative to the healthy controls. And so we're calling this sort of a sort of treatment response networks, right? So it's because it's the difference from the healthy controls. And we can do the same thing individually in responders or non-responders to treatment. And I should mention for fMRI and for structure, we're using as our features, for fMRI, we're using just an amplitude of low frequency fluctuation map, just because we wanted a single map that we put in there. And for structure, we're using a gray matter map. Okay? And so what you can see here is that we get a number of different regions in frontal and thalamic and limbic regions. We see significant differences between pre and post, and we see correlations here in the two groups differently with the scores, and these are cross-validated. And if you look at the within group maps, you see that we get more sort of frontolimbic regions with the responders, and more sort of a there's thalamic thalamus and medial temporal lobe in the non-responders. Another example, cognition-guided fusion in schizophrenia. So here we've got three modalities. We've got again, fMRI, we're using FALS. We've got fractional anisotropy. We've got gray matter maps. Okay? We're, again, using this sort of constrained fusion approach to identify a joint component that consists of 3 modalities that was constrained, it was optimized to correlate with cognitive score, sort of a composite cognitive score. The first thing we did was once we generated this, we did the same thing in an independent data set. It was a slightly different measure, but it was, again, a sort of an overall cognitive score, the MCCB, Matrix Cognitive Consensus Battery, and compared the 2 sets just to see how consistent they were, and actually they were highly consistent with one another. And then we looked at the degree to which we could take the resulting loading parameters from this and predict other domains of cognition in independent cohorts of data. So here you can see some of those results. We've got an FBurn data set, a UNM data set, another COBRE data set. And we also can take a look at these domains individually by looking at separate joint fusion results constrained by each of these individual cognitive variables. And if you look across here, you'll see gray matter and FA are, they're very consistent with one another across, the 4 different domains, which are somewhat correlated with one another, you might expect. But the functional maps are showing a lot more variability, again, consistent with what you might expect. You can kind of see that here in this plot where I'm showing you in red the gray matter loading, in green is the fractional anisotropy loading, and then I'm showing you 2 loadings from 2 of the fMRI components here. And you'll notice that the red and the green are pretty consistent and positive all the way across these different cognitive domains, right? Whereas the functional ones are sort of varying, quite a bit. Okay, and then the last example is looking at autism subtypes. So here we've got, again, we're taking the ABIDE-1 and the ABIDE-2 data sets, and we're combining the data from fMRI and gray matter maps with this autism diagnostic observation schedule, so the ADOS score. Okay, and so we're getting sort of an autism measure that's used to sort of guide the fusion. And so we get maps out, and we check to see that it predicts, again, in different data sets, and you can see it does. And then the more interesting part is if we look at then how well it predicts other symptom-based measures in these sort of traditional subtypes. So we have Asperger's, we have not otherwise specified PDD, we have autistic. And you'll notice that across the 3, we have social interaction is predicted accurately by all 3 of these. And these don't predict one another at all. Whereas NOS, PDD, NOS includes also social communication, and autistic includes all 3, stereotype behavior as well. If you look at these networks that come out, they are also somewhat unique to the groups, where we have FALF-only regions, we have gray matter-only regions, and then we have linked FALF and gray matter regions that are showing up. So this is, I know this is complex, I'm throwing a lot at you, but it's also, I think it kind of helps us, it distills down quite a bit from when you had, like, I've got 3 different data sets, I've analyzed them all separately, and I'm trying to put them together. These are all joint in one single model. Maybe it's a little bit more intuitive, but you do have to get into that mindset and think about it a little bit to get the sense of what it's telling you. So let me go through a few additional models that, because we're far from done yet in terms of figuring out how best, to leverage the data. One might say, well, I've got all these different things I compute. Let's just throw them all in, right, and see what we can get out. And so you can do that. We would, in this framework, we call that Metamodal Fusion. And the way we do it is we take all the features and we further cluster them, and then we treat it as a probability space, and we map probabilities between the features. The nice thing about this is it lets us do either short or long path type inferences, right? So we could look directly at feature M to feature L, or we could look at, how feature L and feature M go through feature N, for example. It's a useful approach. If we just look at sort of the two-way short path information here in a schizophrenia data set, so I've got a bunch of different features over here from just sort of diagnostic information, structural MRI, functional MRI features, including dynamic connectivity, 4D FFT, a bunch of things we threw in there, cognitive scores, symptoms. Notice the symptoms column's blank here. But we do find some negative correlations with cognition. The image-based features tend to be correlated with one another to a degree, although not perfectly. And so again, it puts everything together within a common framework here. Another approach, we can leverage some of the neural network models and look at linking together dynamic connectivity and brain structure. So we know that function rides on structure, but we don't know that relationship. And obviously, the imaging data is very, very coarsely sampled. And so we can leverage basically a learning attention model and a learning alignment model. An attention model would be, just the example is you've got figures with captions, and what the model is learning is if I highlight a word in the caption, tell me where in the image is, what in the image is relevant to that caption. So that's the attention model. The alignment model is used for translation. So for example, English to French, I might go from one word to two. The words might be in a different order. And so what we're doing in the imaging data is we're treating the structure as the image, right? And the function, in this case, dynamic connectivity, we have a time course for each individual subject, which tells us which connectivity state that they're in. And that changes over time, and that's like the caption in the figure. And so we put this model together, and we find very, very strong alignment between function and structure. So this is way significant. It's not anywhere near zero. And in addition to that, we see significant differences. The distributions here are between schizophrenia patients and healthy controls. So here, the healthy controls are on the right side, so the alignment between this middle temporal gyri structural feature and state 3, which I'm not showing you, I probably should have, is stronger. There's a stronger function structure alignment in the patients than in the controls. And that also correlates with a cognitive score here. Okay. We are also often interested in bringing in other high-dimensional data that's not imaging data. So genomic data, for example, or epigenomic data, DNA methylation, something like that. We do that all the time with something called parallel ICA, and I'm not really going to go into all those studies, but essentially, just to kind of introduce what we're trying to do is we're doing an ICA decomposition of two different matrices to maximize independence. And we're looking at maximizing dependence between a subset of those components across subjects. So it's a joint optimization of both separating into features and linking between data sets. Okay? And we can use that approach to take dynamic connectivity states and single nucleotide polymorphism array data and look for essentially common covariation among those in a parallel ICA framework. And what I'm going to do is just jump to the results because I like the, because I think it kind of motivates what we're trying to do. Here we've got a SNP result that was linked to one of the states here, state 1. Okay? We derived the polygenic risk score from the Psychiatric Genomics Consortia data and computed the score on these SNPs. And then what we found was a strong negative correlation. So the more the link between the SNP and state 1, the lower the risk of schizophrenia. This state happened to also be in a separate analysis looking at function, was a state in which healthy controls were occupying much more time, state 1 for much more of the time, during the scan. And the schizophrenia patients were not. They were occupying a different state. So that, although, we've got this sort of partially replicated, it has to be replicated more, right? But it was quite an interesting result, and it was really nice to be able to find this link between SNPs and states, because then we can then start to look at, putative biological functions. Of course, this is pretty far removed. We've got a lot of work to do to tease it all apart, but at least gives us a step in the right direction. And some of the highest SNPs were on chromosome 6, consistent with the PGC results, and included things like neurological disease, psychological disorders, et cetera. Okay. And then the last one is, okay, I'm going to show you a couple slides of sort of, let's, again, I've never, I don't like throwing away information in data, right? And so with fMRI, for example, we've often been reducing it down to a single map, like a FALF map or a component map, and then looking at covariation, right, with another data set. But are there ways that we can leverage temporal information more in fMRI? We're starting to do that with the dynamic connectivity, but we can do more of that. Here, just as a two-step process, we have concurrent EEG and fMRI. We can simply do 2 different group ICAs and then include a deconvolution model to basically map between the time domain in the EEG data and the fMRI data to find links between them. So that's kind of a two-step process where these decompositions were not informed by the second sort of linking step, but it is useful, it is informative. This is sort of an in-between process where, okay, I've got a parallel ICA approach where I've got a SNP array data and MRI data, but on the MRI side, maybe I want to look at age subgroups, right? Maybe I don't want to just treat everything as sort of continuous. And so I can add an additional step in here where I'm sort of decomposing and back reconstructing age-specific components in the context of this decomposition, right? And it lets us then look at age effects, and we can optimize for those effects as well. And then thirdly, well, let's do this with time, right? So we can just take a group ICA here on the left, where each subject is time by voxels, right? And then we've got an ICA on the right, which is basically subjects by voxels, gray matter maps, for example. And we're doing a full group ICA here, getting out resting networks and their time courses. And on the right, we're doing a sort of source-based morphometry. We're getting out a map and loading parameters. And then we can compute a loading parameter like a variable from this data by looking at the correspondence of any given subject's map to the mean map, right? So that sort of relationship, how well do they represent the mean? And so with that similarity measure, we can use that to link it to this data on the right and optimize this, basically in a large sort of analysis that jointly looks at maximizing the link as well as optimizing the independence. Right? Why are we doing this, right? Are we torturing ourselves, right? Well, I think it's useful. The reason, the payoff, of course, is in the end, you have so much more information that you can start to look at, again, all in the same context. It's not done separately. There's actually links between everything. And so here we've got an fMRI map. Because we have and we have the structural MRI map, and we have the link between their loading parameters, but we also have time courses here that we can use to compute a functional network connectivity matrix, right? And we can look at those. This is the group difference between the two. We can predict cognitive scores from those measures as well. Okay, so a few closing thoughts. So I just kind of wanted to say that, okay, more flexible models, kind of that's the theme here, right? I keep getting more and more complex, but more flexible. There's a trade-off there. But it's pretty clear that, that flexibility is kind of important in that, well, so here's structural MRI data, separating schizophrenia from healthy controls reasonably well. SNPs never does well, at separating. But if you combine them, and I don't know why it's cutting off part of the view, but this separation here is actually, trust me, it's cleaner. There's a greater difference between the two. When again, we're using a nonlinear model here. It's basically a deep belief network. And probably all are getting tired of all the papers coming out now because everybody's doing this, right? Everybody's doing prediction and, everybody's doing deep learning and all sorts of things, right? And so here's another example from an Alzheimer's study. We can predict Alzheimer's, quite readily, well over 90%. We can predict if MCI is going to be progressive at about 80%. But we need to get beyond just prediction, right? We want to understand what's going on in the brain. You can start to do that by doing sort of relevance propagation or looking at the layers and trying to map it back onto the brain. I don't think that's enough. I mean, I think I really like basically the network-based approaches, right, where we kind of can see how different parts of the brain interact with one another, which ones are relevant. And so we're trying to do this, and this is sort of a framework that we're developing that tries to sort of unify a lot of the linear decomposition approaches. So PCA, group ICA, parallel ICA, linked ICA, et cetera, and lots of acronyms. But it's all kind of put within one framework, right? So then we can compute a single cost function and compare these different algorithms more directly. But not only that, we can start to extend the blind source separation approaches so that in various ways. So for example, we can create what are called subspaces where maybe we have, like a module in a matrix that we actually, think should be connected, right? And so we can create a model that actually sort of includes that prior information. Or we want to sort of enhance a structural functional relationship and provide that. You can show in simulations, if you get that right, you do much better, right? So the trick is going to be getting it right and or estimating it from the data. And we can also extend these models to add additional layers and look at deep subspaces as well. So basically, we're trying to see if we can map between these things in a flexible way, and also try to predict this the model structure from the data. But it's a hard problem. There's an infinite number of model structures, of course. And so I will end there. Just like to show we try to release all our software for folks to use and potentially build off of. And thanks to many of the folks in my group and collaborators who helped make all this work happen. And thanks for your attention. Do we have time for any questions? Or? Yeah, okay. I know I'm keeping you from coffee, so it's going to be a hard sell, but is there any questions? Yes. You just wanted to get up right now.
I wanted to get up and stretch, but I mean, these are beautiful data, and you've got lots of lovely accumulated data sets where you've got these various features fused in patients and controls. And I'm wondering, I'm thinking about the future and fantasizing about the days where you've got these databases of these fused features, yeah, and then someone new comes along, and what's their likelihood of responding to a particular treatment or not, given the number of attributes that they have, either with structural or functional connectivity. Can you speak to that a little bit?
Yeah, I mean, that's exactly where we're going with this, which is basically as the ends go up, as we get access to more data and we can build these models, you can flip them around and predict from new. And so a lot of the results I showed you, we're actually doing that within our own group, right? So we have extra data sets and we're fitting a model here, predicting over here. Ideally, if we get something that predicts really well or predicts well enough, in a relevant context, then we can basically provide that as a template for people and provide a little tool for people to predict from their own data. And so it's very similar to, providing resting network templates out there, but you just have a few additional steps. And yeah, that's exactly where I think we should go, and we need to see how well this kind of approach would work. I think NIMH is making us submit models too, so which is a good thing.
So thank you very much for the presentation. So usually in machine learning, what you see is that things that work with 100 subjects or 200 subjects, for example, like what you have with the schizophrenia cohort, don't necessarily are the one that work with 500 or 1,000. Basically, sample size affects the choice of methods you make. So what seems beautiful here may not be the best thing if you have less data or even more data. So do you see this in your analysis? Do you see baseline methods, for example, work better in certain settings? Or said differently, also, do you see any limitations in the types of approaches that you're showing?
Yeah, so to the degree, kind of depends on what you're trying to do. So to the degree that we can do this in a way that assumes that there's a common relationship, I think more data will be better, right? In some of the cognitive scores, when we were predicting cognitive scores, we actually had, I think the data set was only about 100 folks, and then the other cohort was 300, and another one was only like 50, right? And it worked in all three of those. But in some other work we're doing, we're trying to use the data to see how should we basically update the diagnostic labels, right? Because maybe there's very heterogeneous, right, within a patient group, and so maybe predicting an average, thing in that patient group isn't what we want to do. Maybe there's subsets, or maybe there's some overlap. Lots of different possibilities there. We've looked at ends, in terms of the number of subjects, quite a bit. We've kind of spanned a pretty big range, from about, I would say, 15 to 20 to 15,000, 20,000. And these approaches work, fine, across a range of those things. It's ultimately going to depend on your effect size and how big those effects are. But because we're leveraging we're using multivariate approaches, and we're leveraging the sample size the samples like voxels, right, or time points, as opposed to trying to estimate distributions along subjects. That helps. It helps stabilize the model a bit, but it's ultimately, nothing's going to work on every case, and it's going to depend a little bit. That's why the simulation framework is useful as well, so we can kind of test that. Yes? What about leveraging the amount of information, including many different types of data? Yes. I think that's really useful. It makes me think also of another thing we're trying to do is basically learn and remove side effects, right? So if, scanner effects, everybody knows we have those, and we try to hope that they're not going to cause issues, but they probably are. And so the more examples we have of that, the better we can learn that. You also have differences in demographics and population and clinical recruitment. Once you kind of get really deep into a particular clinical application, it gets really messy really quick, right? Because the recruitment and the treatment and the all of the protocols, right, very detailed and very different, right, from site to site. So I think capturing that variability is going to be really the first step, so that we can. Maybe it'll help us say, well, these are the 3 things that we should try to make sure is consistent, right? And these other things are less important, though we know they're variable, but we can control for them. So that's my hope, is that we'll be able to do that sort of thing.